Potential new gene editing tools uncovered. This is a new today's article by University of Texas at Austin. Few developments have rocked the biotechnology world or generated as much buzz as the discovery of CRISPR-Cas systems, a breakthrough in gene editing recognized in 2020 with a Nobel Prize. But these systems that naturally occur in bacteria are limited because they can make only small tweaks to genes. In recent years, scientists discovered a different system in bacteria that may lead to even more powerful methods of gene editing, given that its unique ability to insert genes or whole sections of DNA in the genome. New research from University of Texas at Austin dramatically expands the number of naturally occurring versions of this system, giving researchers a wealth of potential new tools for large-scale gene editing. Other scientists had identified clusters of genes that use CRISPR to insert themselves into different places on the organism's genome, dubbed CRISPR-associated transposons, Cas for short. Earlier work has shown they can be used to add an entire gene or a large DNA sequence to the genome, at least for bacteria. And now a team led by Ilya Finkelstein and Klaus Wilk at UT Austin have expanded the number of likely casts from about a dozen to nearly 1,500. They published the results this week in the journal Proceedings of the National Academy of Sciences. With casts, we could potentially insert lots of genes called gene cassettes, encoding multiple complicated functions, said Finkelstein, Associate Professor of Molecular Biosciences, who conceived and headed the research. Among other things, this opens up the possibility of treating complex diseases associated with more than one gene. Kindly support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box. CRISPR researcher and Nobel laureate Jennifer Doudna has predicted casts will be a critical element in expanding genetic engineers' toolkit, making it possible to introduce any change at any genetic location in any organism within the decade according to genetic engineering and biotechnology news. Using the Stampede-2 supercomputer at the Texas Advanced Computer Center, TAC for short, the team combed through the world's largest database of genome fragments from microbes that have not yet been cultured in the lab or fully sequenced. Without the resource of TAC, this would have been impossible, said Wilk, professor and chair of the Department of Integrative Biology, who led the data engineering part of the project. He estimates that if the search was run on a powerful desktop computer, it would have taken years. Instead, the one of the university supercomputers the final analyst has completed within a few weeks. 3 graduate students, James Rybarski, Kuwang Hu, and Alexis Hill, worked full-time on various aspects of the project for nearly 2 years. Finkelstein said, "The term for this is bioprospecting. It was like sifting through a lot of silk and junk to find the occasional gold nugget." The UT Austin team found 1,476 new putative casts, including 3 new families, doubling the number of known families. They have already experimentally verified several of these and plan to continue testing more. Ultimately, Finkelstein predicts most will turn out to be true casts. If you just have a handful of casts, it's unlikely that you have the best ones in existence, Wilk said. But by having more than 1,000, we can start to find out which ones are easier to work with or most efficient or accurate. Hopefully there are new gene editing systems that can do things better than the system we had beforehand. In the short term, Finkelstein said many of these new systems should be adaptable to genetically engineering bacteria. The long-term challenge, Finkelstein said, is to domesticate the system to work in our cells. Finkelstein said the Holy Grail is to get this working in mammalian cells. So this is on phys.org by University of Texas at Austin, today's article. Please leave your comment. Thank you for your support.